Hello viewers, today we are going to discuss about the topic moving boundary electrophoresis. Electrophoresis means movement of charged particles in a medium under the influence of electric field. It is of two types, zone electrophoresis and moving boundary electrophoresis. In zone electrophoresis, the separation is carried out with the help of supporting media like paper or gel. In moving boundary electrophoresis, the separation is carried out without any supporting media. Moving boundary electrophoresis is a technique in which the movement of charged particle occurs in a homogeneous solution. It is also called as free electrophoresis. Next, principle of moving boundary electrophoresis. This technique was made based on the differential migration of ion under an electric field. The interface between the migrating ions and the buffer solution is observed as a boundary, hence the name moving boundary electrophoresis. Next procedure. Separation is carried out in a U-shaped tube. The bottom of the tube is filled with the buffered solution of macromolecule. Pure buffer is added above it. Whole cell is kept in a temperature bath. When the power is switched on, the sample which has the positive charge will move towards the cathode, whereas the negatively charged ions will move towards the anode. pH is selected that all the molecules should carry a net positive charge or net negative charge so that the ions moves from the macromolecule to the pure buffer solution and forms the boundary. Next, applications of moving boundary electrophoresis. This technique is mainly used for the separation and purification of proteins, diagnosis of diseases by separating blood proteins, and we can understand the behavior of macromolecules under electric field. Advantages of moving boundary electrophoresis: simple setup, effective for separating small molecules, and the limitations are it is not suitable for complex mixtures, limited resolution compared to other types of electrophoresis. In zone electrophoresis, the separation is carried out in a semi-solid or solid medium, and in capillary electrophoresis, the separation is carried out in capillary tube. While comparing to other types of electrophoresis, it is the simplest method of separation. Moving boundary electrophoresis is the oldest method of electrophoresis, and it is very useful to understand the basic principle of ion migration. Thank you.